So, a very good afternoon. I, Sharanya Trivedi, heartily welcome all the delegates and participants who have joined this FDP program exclusively focusing on bioinformatics, named Data Driven Biology, exploring computational tools for genomic and structural insights, organized by the Department of Biotechnology of the Neotia University. We have very recently organized another faculty development program of 7 days that aimed on different areas of microbial biotechnology in the previous week. And today, as we begin the FDP of a tenure of 5 days, that is from 3rd March to 7th March, scheduled from 2:30 to 4:30 p.m., we expect for a substantive participation and procurement of knowledge on bioinformatics. Bioinformatics, the interdisciplinary field that combines computer science, mathematics, and biology, has revolutionized the field of biotechnology. By leveraging computational tools and methods, bioinformatics has enabled researchers to analyze and interpret vast amounts of biological data, driving innovation and discovery in biotechnology. As bioinformatics continues to evolve, we can expect even more innovative solutions to emerge, transforming the field of biotechnology and improving human health and well being. And for this comprehensive training on bioinformatics, we have our esteemed course coordinator, Dr. Ranujit Kumar Sarkar. Dr. Ranujit Kumar Sarkar is an accomplished academic and researcher. I mentioned he is an immense specialist in the field of bioinformatics with nearly 20 years of teaching experience and research. He has completed his MSc in bioinformatics from the University of Pune in 2004. He has done his thesis on bioremediation of low density polyethylene LDPE. Dr. Shorkar has authored over 15 publications in international journals and delivered talks at numerous conferences, earning several awards for his contributions. He is a recipient of prestigious scholarships, including the ICCR, Government of India, and the G.N. Ramachandran Fellowship. Dr. Sharkar, in this training program, will provide us a thorough insight on navigating databases from sequence retrieval to structural insights, protein family selection and efficient data extraction strategies, advanced blast strategies for domain conformation annotation, genome-wide analysis using phylogenetic tree construction. So, without any further delay, let's begin with our very first day of this FDP and dive into deep knowledge and detailed understanding on the advanced technique of bioinformatics. Over to you, sir.
Thank you, ma'am. I hope I am audible.
Yes, sir, you are audible.
Thank you. Thank you very much. Uh, Let me share my slides. My slides are visible.
Yes, sir, it is visible.
Okay, thank you. Okay. I welcome all of you in this faculty development program on various bioinformatics resources. So today, our focus is protein sequence databases and protein structural databases. So today, basically, we will discuss the basics of databases, how databases are collaborated among each other, and how we can use various terminals for retrieval of sequences. And then finally, the main objectives of today's session is to means solve various research-oriented questions. How those are faced by the scholars or means researchers, that how to retrieve a correct sequence one. So if we go for PDB databases, you have seen that for the same protein, you are getting so many hits. So which one is the correct one? How to select that one? What is my objective? And according to my objective, are there the correct protein sequences? If the sequences are available in more than one number, then which one would be the better one? If we do not have any sequence listed as per our requirement in the databases, then what to do? So all these type of questions we will try to resolve today so that we can be benefited from the bioinformatics databases in our research. So let me allow to start today's session. Thank you. So if we think about bioinformatics, database is the first question that comes to our mind, that what are databases and why biological databases or bioinformatics databases are required. Okay, so if we think about databases, you know that often the database, the terms is abbreviated as DB. So shortly, most of the time we call them DB. And it's a physical storage, electronic storage of some information. And what is information? The information definition of information is very critical. What I mean by that, that a piece of information should be retrievable. Okay, so when we deposit something, when we store something, and that should be retrievable, and that can be analyzed further, then only we will call that piece of information as data. Otherwise, that is not data. So that is the clear means the difference in between a piece of information and data. So data must be retrievable, and it should be used for the analysis. Okay, so that is the data. So when we deposit data in some physical storage in an electronic form, so that is termed as databases. So you see that this cartoon will explain you that actually how our databases are uh, means working. So if we are the clients, the word we are using client, so we are requesting the physical storage, that is database, for some data. And how we can approach the databases for data? We can write some algo, we can write some code or query, you can say, and that query will be translated to the databases and we can retrieve data accordingly, right? So these are the databases. Now the next question comes that then what are the requirement of biological databases? Okay, so if you think about biological databases, then first you need to know that what are actually biological data. You know that we have sequenced so many organisms genome so far, and including human, and then the genome sequences. We thought that the genome sequences will be the end of our research, but now we understand that it is just the beginning. What do I mean by that? I mean that if you have the genome sequences, then the next course of action is there that to make the catalog of genes. So what are the physical locations of gene sequences? What are the start position and what are the end position? And of course, the regulatory regions of those functional units. So this all together give you a challenge that how you can organize the huge data and how you can retrieve and efficiently analyze these sequences for these insight informations. So say if we do not use the databases, electronic forms of databases, if you deposit, if you store the data in a hard copy, if you think about human genome, you know that the size of the human genome is 3 billion base pairs, right? So if you write ATGC, 3 billion ATGC on means paper, you can imagine that that will be 20 equal volumes of Lellingers. Now, in 20 equal volumes of Lellingers, what is written only 80 and GC. So, is that a data? No. As per the definition just we discussed a few minutes back, it is not data because you cannot means use this information for further analysis. Like I said that the identification of any gene regions, comparison of gene sequences across the species, finding out mutations, and so on. So that's why it has to be deposited in electronic forms. So now you have got the flavor that how why the bioinformatics databases are relevant. Not only that. So all over the world, the scientists are means continuously they are generating data. So if we can maintain the data in a high quality, retrievable manner, then immediately that data, that information can be accessible to the all the researchers. So that is the beauty of this information technology. Now, what are the challenges? It's very much okay that we are interested in depositing data in an electronic copy. That's not the problem. The problem is that the size of the biological data and complexity of the biological data, because it has to be retrievable, right? So that is data all about. So now the complexity means the biological data is text data as well as it is a three-dimensional data and lots of patterns are there inside along with the bibliographic data. So it is very much complex as compared to the non-biological data, right? So how the data can be arranged in a database, that is a still today it's a challenge for us. So if we think about the traditional biological data that is working so far, you will see that they are flat file formats, and these files are actually composed of fields and records. So what these three terminologies means, briefly, I will not go in details, but briefly I will tell you that fields are the single piece of information. So means the name of the protein, the length of the protein, okay, mutations of the protein. So all these things are some fields, specific fields of information. So when a particular field, a piece of information is tagged with a name or ID, that is the record. So that is the complete set of one field. So that is a record, and a number of records actually a file contains in that way. Okay. So, biological databases, you know that there are various types of databases like the primary databases, the secondary databases. There are so many primary databases. Already you are aware of this fact that there are NCBI, there are UCC Genome Browser, there are MBL, and so on, DDBJ, and some structural repositories. So will not go in details of that, but many times we are confused that how to use these databases, how databases are working, and should I go through each of these databases to find our protein of interest or not? So that questions we may solve now. So you see that how the database is running. Okay, here you can see that we are the submitters, okay, the researchers, the research scholars, okay, students, they will submit genomic sequences, gene sequences, patents, and various updates because they are working on that. How they can submit? So there are some submission tools. So they can use some submission tools, and through that they can submit this piece of information into the core databases that is termed as production DB. Okay. Now this production DB can receive databases from some other archives also. Okay. It can get information from other archives, and then This data actually is categorized into several classes, into several service DBs. Like you see that they are termed as service DBs. Now, the end user, when we are retrieving sequences, the same scientists or scholars, we now become the end user. Okay, and these end users can write some query, and this query can be submitted to some service tools. Like you know that in case of GenBank, it's the Antres. Antres is the service tool. In case of EMBL, it is the SRS. That means sequence retrieval system. So you write your means query and submit it to service tools. Why it is submitted to service tools? Because actually we need to write some complex SQL queries. And you know that the SQL queries are a bit difficult, bit complex. So we, the biologists, we do not may not know or may not like writing the SQL queries. So it's very difficult for us then it will become to retrieve sequences from the databases. So there is an intermediate program named service tools, which actually take our query as input, and then that input can be means explained to the SQL query system in details by these tools, and which in return will be submitted into the service DB, and we will retrieve the sequence of our interest or any data of our interest. So that's how actually our database system is working. Now, if it's so complex, that means there are several databases, they are interlinked. Okay, so this slides actually explaining you that and solving your query that should I go through all the databases? No, it's not required actually. So you can select means terminals of your convenience as per the requirement of your data. So the primary databases, basically, the primary databases and major secondary databases, they are interconnected. So if I search through one terminus, then I can retrieve data. If that is not present in the database I am searching for, that may be present in other databases, and I can get that. So through that terminus I am using. Okay, so basically we are focusing on sequences both for DNA and protein, then proteins domain family. We try to means analyze mutations, polymorphisms, proteomics, 3D structures, metabolism, and so on. Okay, so that's we are going to see. So see, this is the uh, beautiful things. So this you all of you know this is the NCBI terminus. We have used NCBI terminus for searching this data. Now here you can see the DB source. Okay, DB source, and you can see it is Swiss prot. Okay, it is Swiss prot. That means that this protein we are searching, it's not present in NCBI GenBank. So, but we are using NCBI Bank. So, the terminals actually retrieve data from Syspro databases. And that's why in the DB source field, so these are the field. So, just a few minutes back, I was talking about field. Okay, so locus, definition, accession, keywords, these all are fields. Okay, and these fields are very much conserved for all the files within the same databases. That means the fields will vary from one database to other, but the billions of files that is present in a single database, that will remain exactly same. Why it is so? Because many times actually we need to write some programs like using Perl script or other languages to manage the data from number of files. So that's why if these fields are you can say not structured or it varies, then it will be very difficult for us. Hello, can you mute yourself? If not any question, can you kindly mute yourself? Evelina, kindly mute yourself. Okay, sorry for the inconvenience. Okay, so where we are, so see that we have used NCBI terminals in these cases, and we have got the information from sysprot. That means that you not to be bothered about in which database your protein might exist. We do not need to be bothered about that. So any terminals as per your convenience you can use, right? Now see that just give me for 1 or 2 minutes only. I will explain you the file and record concept because it is essential for further means exploring. That's why I'm giving 1 or 2 minutes here. You see that this is the locus field, definition field, accession field. Now the locus field actually you see that this is the accession number of the file. This is the length of the protein, the type of the molecule, that is amino acids. It is a linear molecule. It's from primates. The PRI indicates it's primates, and this is the date of submission. That means it's altogether the summary of the file that is there in the locus field. So in any file, if you go open in the LCBI GenBank, you will see that there will be a field called locus. It will be the first field, and accordingly, all the fields will be very much conserved. Okay? So that's all about fields and records. So locus is a record, then this is a field, and with this information, it is termed as record. So there will be number of records like locus, definition, keywords. So all together, it's a file. So the record can be single line or multiple line. Okay, so let's focus on sequence data. So now you know that in our sequence database files, we are basically interested in sequences of codes. Then accession number. Why accession number? Because it's a unique number. You know that there are billions of files in the databases. So if you use any keywords or gene name or protein name to search a files, you will get hundreds of files there. So which one actually you are interested in, it becomes very difficult and confusing for us. So if we have the accession number, very uniquely we can retrieve that file from the databases. Then the taxonomic data is also important. You know that the when you are doing some research, then narrow spectrum and broad spectrum plays a critical role in your research. Okay, so that's why taxonomic data can be crucial for you. Then the references is also an important part. You can understand the means quality of the data from reference sections. Then annotation and curation is the one of the most important part. Okay, so that's why you will see that today actually we are moving towards uniprot databases. So all of you might be using so far the NCBI databases, but today actually I will discuss with you the UniProt databases mainly because it's very informative. It's a vast databases and it can means solve lots of your research questions. It means UniProt today we will see that. So then there are some keywords and cross references for further technical reasons. Okay, now you can see that the thing I was talking about, although it's not much visible, but if you get time, you can go back to the different databases like EBI, NCBI, and NIG, and you can see that these formats really they vary from one database to other. For the same protein, you will see it will vary from one database to other, but for the same database, it will be highly means exactly same, you can say. So now these are the some list of databases, that is secondary databases, that is ProSite, it's a patterns or profile databases. In the day two, we will discuss about ProSite and ProDOM, PFAM, you can see blocks, scope, CAT. So these are the some secondary databases, very informative databases. So today we will focus our discussion mainly on the primary resources of protein databases. So let's begin. So if you start with the UniProt databases, many of you are familiar with these home pages. So of course, we will go for the hands-on session. But before that, it's good to have a fair idea about the database and its organization. That will help us in details to learn the hands-on session uh, that will be followed by this session. Okay, so let's start. So now you see that there are four different sections. Okay, so these are proteins, uniprot knowledge base, then species proteomics, protein clusters, and sequence archive. So there are four different sections. And out of these four different sections, you can see that the Unicode knowledge base has two subdivisions. If you have noticed, you can see that there is a golden color one that is reviewed, and silver color will be marked as unreviewed. That means the reviewed part is from Sysprot and the unreviewed part is from TR-EMBL. TR actually stands for translation. Okay, so translated sequences. Now you can see there is a huge difference in the number of protein entries in between these two databases. So all the proteins basically we search from UniProt knowledge base. If we are interested about some organism-specific proteins, then we will go for species proteomics. Say if I am working on leishmaniasis, or I am working in malarial vivac, or say I am working on some model organisms, then I might take interest in species proteomics. If I am interested about some group of proteins across the species, then I will be interested in protein cluster databases. And some sequence archives, if I am not concerned about the quality of sequences and structures, but I need some sequences that is similar to my target sequence because that is very unique. I'm not getting data in the known domain. So then I will go for sequence archives simply per knowing that there might be lots of errors, there might be lots of discrepancies. So I will keep that in my mind while interpreting my results and while writing my manuscript. So basically, we will focus on Uniprot knowledge base today, and this is the region where you can get your protein. Okay, so let's summarize a few of the questions that always we get from the scholars or supervisors that how can I get the structure of my protein? Is the structure of my protein known? How can I get that? So many times what we do, we directly go to the PDB, protein data bank. You know that protein data bank is the structure database. So what we do, we go to PDB databases, we search the PDB database through some protein name or keywords, and we end up with a series of structures. Okay, and we get confused that which one actually the structure required for me, I don't know that. Okay, and which one to select. So selection of correct structures will be very crucial because that will influence your research a lot, right? So this is a very, very important question that how I can get the correct structure. Correct means structure of your requirement from the databases. So today we will see that how we can navigate from the UniProt to PDB, and that will be the best approach. Okay, so you can uh, move from UniProt to PDB for the selection of the correct protein structures. So you might not be confused about that. So let me some time shortly we will start the discussion regarding this. So now which structure I should select? So you will see that the same structure is there. Okay, now what are the criteria that might be considered for getting a good quality structures? So that is another area of concern, right? Now the third question is that how my protein interact with other proteins, how I get that information that I have a protein might be, and most probably that protein is involved in a networking, okay, network of proteins. So the function of my protein will be influenced by those networking proteins, and simultaneously my protein also will influence those. So how can I get those informations? Okay, how many domains are there in my protein? Is my protein having any domain? What are the domains? What are the positions of these domains? And the structural insights of those domains. How can I get that? Okay. Are there any disordered regions in my protein? Why you are interested in disordered regions? Because if there are some disordered regions in the protein, you know that these disordered regions are might means influence negatively while you are developing your model, like you can use homology modeling modeler or something like that. So you have to get the disordered regions, and you have to remove the disordered regions from your sequences, because these disordered regions might means interfere with the quality of your structure. So that's why these are the some of the important questions. I could not include all the questions, but few of the questions always I came across, I tried to enlist for you. So let's see how we can solve this one by one, and definitely we will practice it at the end of the session. Now see that UniProt results. So I know that you are familiar with this output. So if I search for a protein sequences, you see that here you can see that this is the way how UniProt will give you the output. Say I have searched for a protein that is EGFR. Okay, I have searched for EGFR, and the result is shown in this way. So this is the unique ID, that is the UniProt ID. Then you can see the entry name. So all these are EGFR. But you know that these EGFR are from different organisms. Okay, you can see the same protein EGFR belongs to different organisms, and you see that the length of the sequence are also varying from one organism to other. It is varying, right? So these are the means output. Now you see that another important field that you see, this is golden mark, okay, and the last one is not. Okay, what do we mean by that? I mean that these proteins are reviewed protein. These are reviewed protein. So there very lesser chances are there that there will be some error. Okay, but this one is a non-reviewed protein. So if I need to select the protein sequences from human, obviously now you know that this will be the better one. Okay, this will be the better one, but there are some several other criteria also. We are going to look into that shortly. Okay, so these are the some primarily. the output entry list. After searching, you will get that. Now you see that in the section here, you can see a list. Okay, the functions, the taxonomy, the subcellular location, the disease and variant, the post-transcriptional and transcriptional processing, the expression, interaction, structures, okay, family and domain, sequence and isoform, and similar routines. So I will explain all these sections to you before going to the web for the retrieval from UniProt. So here the function you will see, I know that you are more familiar with NCBI. Now, if you go through NCBI protein, I am sure that such a comprehensive function section you will not get there. That's why UniProt is really unique in terms of research. Okay, so if I am a researcher, it will be a good one to use UniProt if I can manage its different tricks and techniques. Okay, so now you see that this functions is also supported by the respective papers. You can see these are the PubMeds. Okay, these are the PubMed ID. That means this function, this function has been derived here from this publication. So if I am working on this area, okay, then I can go through these papers and I can get the insights of the functions in details, right? So I need not to go for searching the research paper and end up with some non-relevant research papers in comparison to my requirement. So that is the high chance if you do not use UniProt. Okay, so now you see that all the functions, not only this one, all the functions are supported by respective research paper. That is the unique data representation in UniProt. So the functions, if you are working on a protein, you can be expert by going through the each and every research manuscripts, the publications that has been published in that area. Okay, so this is the beauty of this one. Now you see that the subcellular locations. Okay, the subcellular locations, the where this protein is actually present within your cell. So these are the uh, organelles where this protein is available. Okay, so the location, of course, you know that you can predict the subcellular location by using various tools. But here also, you can get the confirm information about the locations within your cells regarding your protein. So your protein is present in cell membrane or endoplasmic reticulum or Golgi apparatus that you will get here. And again, see, these will be supported by the respective publications. This will be supported by the respective publication. So I am working in this with this protein, and I am interested in its mechanism of function in nucleus. So I can go through these papers. If I am interested in cell membrane and both, so I accordingly I can make my preparations. So that is the subcellular locations, and you know that this is a very important information in terms of your proteins. Okay, so now another area of interest is the disease and variants. That this means if you use any databases, you will get very less information as concise as is in UniProt. You see that this segment will tell you that what are the mutated forms of the proteins and the details about that. Again, that is supported with your publications. Okay, so the changed versions, like say if you are working with sickle cell protein, and you will get the variants of these and where the changes are there, and because of that which disease you are getting. So all these informations you will get in the disease and variant section. Not only that, you will get the details of those research in that publications. Okay, so all the publications will be supported. Here you can see these are the publications in lung cancer. You can see this protein involved in lung cancer, and these are the three publications regarding that. So this protein again influence in neonatal and fractional and inflammatory syndrome, and you can see that there is a it is supported by another publication. Okay. So now the next one, again an important one, is the post-translational modifications. It is the post-translation. Just a minute. Just a minute.
শোনা যাচ্ছে না।
Just a minute. Sorry. Okay. Now it's fine.
Yes, sir.
Okay. Okay. So now you see that this is the segment of post-translational modifications. See how many post-translational modifications have occurred within my protein. You see that there is glycosylations. Okay, so this is one type of modification. So there will be many. I will show you. So this is glycosylation. What is the idea of that modifications, and what is the positions, in which positions of my protein? So if this is the length, this is the scale of my protein, and then in which position actually this changes has occurred. Like you see that this is the 56 positions. Okay, glycosylations has occurred. Then at 73 positions, glycosylations has occurred, and also at 123, 128th position, the glycosylation occurred. And this report that has been published in these publications, that is also marked here. So if you are interested, if you are working in this area, you can go through these publications and you will get the details on that. Okay. So now next, you see that the interactions, the different subunits, and how these subunits interacts with other proteins, and what are the mechanism of that. So that also you can get through from these interaction sections. Again, all the sections in UniProt that is supported by the respective journal. Okay, you see all the information, that whatever information it is marked here, you will not have any query about that, because you will get the relevant publications for that. Okay, so that is the beauty of this uh, databases. Now you see that the point I was talking about, that structure. Which structure? Are the structure known? If I am working with a protein, is the structure of that protein is known? If it is known, then which are the structure I should select? Now you see that if I move to the structure sections, you see that the structure is known, and there are several structures of my protein that is similar with present in the databases. Okay, in the PDB databases. Now the problem comes that which one to select, right? Which one to select. So that part we will discuss while we are going through UniProt databases, right? So these are the questions. That means that if I'm working on a protein, it's better to go to PDB via UniProt, right? So via UniProt, we can go to PDB and we will get the more accurate proteins of our interest. Now you see that the families and domains. So if there are some domains that is present in my protein, so I will get that here in this family and domain section. Here you will see that there are some domains, okay, and its positions. There's some domains and its positions. Okay, so that we will also discuss. Now you see in the databases, another important aspect is the understanding protein variants and their importance. Okay, protein variants. That means the isoforms. Isoforms is present in the protein databases. So let me tell you what are isoforms. You know that the one gene, one protein theory does not exist today. So earlier, we were under the thought that one gene codes for one protein, right? So this theory does not exist today. Why? You know, because of splicing, one gene can produce different protein, completely different protein. Functionally, they are different. Okay, from one cell to other, from one tissue to other tissues. So that is the main important part that your gene is same, okay, but your protein can be different. So if there are different forms of proteins, they are reported under the section of isoform. They are reported under the section of isoform. That means the isoforms are the group of proteins which are alternatively spliced, so they become variants and their functions can be different. Okay, so they are very, very important. So these isoforms basically arise due to alternative splicing, alternative promoter uses, or ribosomal frame shifting. So these are the various points that we should think about regarding the isoforms. Now, these isoforms may differ in sequence, functions, and cellular localization. As I told you earlier, that because of alternative splicing, mainly, we are getting these isoforms across the tissues. Okay, so that's why there is a high chance that their cellular localizations will be different, and in different tissues, their functions also might be different. So these isoforms and its variants are very, very important, and it's very hard to detect in other databases other than UniProt. Okay. So these are the information that you can get, that how many number of isoforms are there, what are the canonical sequences. That means this is the reference sequence. Which one is the reference sequence means which with respect to which protein actually you are saying that they are isoforms. So that is the actually the canonical sequence. Then the alternative sequence is the length of its molecular weight, the experimental evidence, and computationally mapped isoforms are also there. So if it is computationally mapped isoform, that will be mentioned. So you should be careful if you are going through computationally mapped isoforms, because you might get some error in this cases. Okay. So these are the significance of isoforms because they are having functional diversity. Okay. So they will have disease associations with them. They are very much important for drug targeting, and to understand the evolutionary insights, the isoforms will play a crucial role. Okay, so these are the similar proteins that you can see in the Prince UniProt databases. Okay, so now we will move to protein data bank. So before that, we will see that we'll stop our presentations here at this point, and we will go through UniProt databases for the hands-on session. Then we will again come back to this point, and we will start with protein data bank. If you have any queries, you can ask me at this point of time. Okay, so if you are having the laptop with you, or if you are joining through laptop or desktop, so you can try with me also. Okay, so to access the UniProt, you just go to the web, and simply you can type UniProt database, and you will get the uniprot.org. Okay, www.uniprot.org. Now you see that, see, this is the UniProt database. Okay, this is the UniProt databases. Okay, so I hope all of you are able to see this one. So from here you can get the statistics. Here you can see the blast tools like NCBI. It's the same blast tools you can use here also. So this is the segment where you can find your protein. Say, for the time being, you are interested to find your protein, that is EGFR, okay, or any protein. If you like, you can search that. Okay, so you can search any protein of your interest here. So see, here we search for EGFR. So I will show you the same protein and along with the different protein. Okay, EGFR. So you can type EGFR. So these are the four segments, okay, that is here. So Uniprot KB, Unilef, and all the some human disease databases, and also included there. So basically we will go for Uniprot KB. So if we search to Uniprot KB, so we can get these sequences, right? So just I showed you these pages only earlier, right? So these are the unique identifier number. What are the role of the unique identifier number? So after the primary search, if you think that these are the proteins that is important for my research, so you can take a note of these ID details. So you need not to save all this data. Okay, rather you can save the UniProt ID. And later on, using this UniProt ID, you can again go to the database and you can retrieve your data of interest, right? So that's why I'm showing you. See, this is the UniProt ID, right? Now, if I search the database, say, through only say P0C Y46. So let's search that one. You will see that this file, just I showed you, not this file. I opened this file. You will get the same file only. See? So by using the unique ID, this is the beauty of unique ID, that using this one, you can retrieve the single file, the file of your interest. Otherwise, you see that when we just used a name, that is EGFR, see, you retrieved 18,000 plus results, right? 18,000 plus results. Okay. Now, out of that, you see that 1,080 proteins are reviewed protein. Okay, from cis-prot. So these are the reviewed protein. This symbol indicates that they are reviewed protein. Reviewed protein means you can rely on this data. They are high quality data. You can use this data in your research. So if you submit a manuscript, your reviewer definitely will ask you regarding this segments that your sequence, you have taken the sequence from the reviewed or unreviewed sections. If you take the protein from unreviewed section, then it will be very hard to convince the reviewer because lots of questions then they will ask. Okay, like you see that the most of the proteins are from unreviewed sections. Now, if you have means epidermal growth factor, you have searched. Now you see that they have listed organism wise also. See, from human, there are 525 protein of the same protein, that is epidermal growth factor. That's what actually I was talking about, that how difficult it is to get the correct protein structure in the PDB. So we will see that how we can move to PDB from here with higher accuracy. That tricks I will discuss with you. Okay, so here you can see that from human it's 525, from fruit fly it is 347, from mouse it is 255, and so on. Okay, there are so many organisms that is not listed. The main organisms has been listed here only. Now, taxonomy, you can filter it by taxonomy if you need that. I require some proteins from this two to three organisms only or five organisms only. You can add some search fields and you can take those taxonomics name, okay, and you can search that. So your output will be filtered out and you will get the protein from your organisms. It might be you are interested in a few organisms only, few species only. Say you are working on a protein, you are targeting the human protein and you are interested to see that how it behaves in mouse, rats, and bovine also. Okay, so you might be interested in that. So in that way, you need not to go through these 18,000 sort proteins. You can simply filter your output, right? So you can group it by also taxonomy, enzymes class, and all these things. And now you might be interested about some proteins that must have 3D structure. Say your research criteria is that you are working on 3D structures only. Okay, so you see that proteins having 3D structures is only 4,336. Okay, 436. That means out of these 18,000 plus odd results, only you have 3D structures 436. So now if I am interested in 3D structures related information, so my research will involve definitely the 3D structures, then I can select this one. So it will be filtered out. Then you see that other than the full length 3D structures, there are 5,000 odd active sites are there. Okay, 5,000 odd active sites are there. I might be interested in various active sites. Then I might be interested in these sections, the active sites. Okay, then you see some protein existence, that is predicted proteins are there, homologies are there. Okay, so these are some uh, other criteria. Okay, some sequence length. So some small sequences you can see 1 to 200 sequence length. These are the number of entries. 200 to 400, these are the number of entries. So you can see that there are only few protein lengths are there, right? There are only few protein lengths. You see greater than 800 base amino acids are only 7,000. So how many of this 7,000 will be experimentally verified structure? That will be the question right now, right? So in this way, you can filter from these sequences. So say I am interested in reviewed sequences only. So you see, I have filtered that out. Okay, so I have filtered the unreviewed segment. You see, the unreviewed segment already filtered out. So out of the reviewed structure, now you see that from human there are 378 structures, from mouse 203, from rat these are the structures. Okay, now another interesting thing you see that only 3D structures 431. Earlier it was, if you remember, it was 436. That means only five structures were predicted. So others are actually resolved structures. So active sites, now you come to this point, active sites, you know that 5,000 plus active sites are there. And now these reviews are only 258. Okay, so it's a huge reduction, right? So that means these 258 active sites are there in the databases that are reviewed and reliable structures you can rely on. Okay, so these are the some other statistics you can go through before you going through a particular entry. So before means going through a particular entry, all these must be carefully reviewed in the left-hand sections, and then you select your important protein. Okay. Now see, I am interested in say mouse epidermal growth factor. Okay, so if I go through mouse, I can see that here is only one mouse. Okay, but mouse there are 203. Might be most of them are say not full length structure, the active size. So let's see this one. What happens? So this is a mouse epidermal growth factor proteins. So this is the epidermal growth factor proteins. And this section will summarize your file. What they are saying, you see that they are having amino acids of 1210. So 1210 amino acids are there, right? So protein existence, evidence at protein level, right? Annotation score, it's a very, very important one. You can see annotation score 5 out of 5. That means it's a highly annotated and reviewed protein. So if you select this protein in your manuscript for study, I hope you will get lesser questions and reliable questions from your reviewer. Okay, so from mouse, if you are interested in epidermal growth factors receptors protein from mouse, so this might be your protein of interest. Okay, now what we are talking about that you might be interested in its function. So what are the function it performs? So what are the function of this protein? The details functions you will get from here along with the, see, this is the PubMed. So just open it in the new window. Just right click it and open in the, see, this is the literature. Okay, so this is the abstract of your protein. So by going through the abstracts, if you feel that you need further study, so you can go for the journal. Okay, you can go for the journal. So these are the these related uniprot KB entries. So we are not discussing that right now. So this is the information actually I wanted to share with you. Now you see that these are the functions. So if you can decide if you want to mutate the protein, you want to explore the changes in function, how it affects the mouse. So this will be a good one. Okay. Now, what is its catalytic activity? What is the catalytic activity of this protein? So these are the catalytic activity you can go through in details and how its activity is regulated. Okay, along with the details mechanism in the paper, you can get its activity regulation, how its activity, its signaling is controlled. Okay, so mechanism of signaling. So that is very important information. Without going through a large number of research paper, you can easily get it here. Okay, now come to the binding sites. Okay, so what are the binding sites? You can see the binding sites, active sites, and other sites. So all the information you will get it here. So you see that it has how many binding sites? It has four binding sites, right? So now you can realize how important this information is. Okay, so it is a binding site. You see the binding site sequence pattern is this. Okay, lysine is there. Okay, glycine is there, serine is there. Okay, so that is the sequence at this binding site. And what is the position? The position is 722, 728. Okay, 722, 728. And what binds here? ATP. What binds here? ATP. And if you want to details of these binding mechanisms, everything if you want to know, you can get here. Okay. Now, how they become different with other databases? It's very difficult in other databases to find or summarize this information. Right? It's very difficult. Why it is difficult? That means this information is not categorized in this way, like that is in case of Uniprot. You see, now say this is the active sites. Okay, you see the active site sequence is D. Okay, it is the D. So D is aspartic acid or glutamic acid. Can you tell me? Can anyone tell me what is D stands for? It's the aspartic acid or
Aspartic acid.
glutamic acid? Aspartic acid, right? So that's why you see that we always need to remember the single letter code because in bioinformatics domain, mostly we actually play with the single letter code. Okay. So all the tools, most of the tools will play with single letter code. There are a few tools that can means accept the three-letter codes, but mostly it's the single letter code. Now see that what is the position of active sites amino acid is aspartic acid. It is at the 839, right? 839. And what is its function? It is the proton acceptor, right? Now see, you can design lots of experiments from here. Okay, if you are a researcher, so it is the means site of your interest. So these binding sites, these active sites, say I am interested to block this protein. I am giving you an example right now. Okay, so what we do, we design some molecules that will means I will make docking of those molecules, right? So these are the docking simulation studies you carry on, right? You carry out, okay, all these researches. Now you see what will happen. The informed researchers, and if you ignore this side, so if you take this protein, if you means do some docking analysis by identifying lots of ligands molecules, and you report that yes, my molecules is binding here, and this is the binding energy, and all these things you have done excellent work. But if your reviewer asks that your binding is other than the active site, then how you will justify, isn't it? So that means you have to be very careful about these binding sites, active sites, and all these features. And you have to see when you are docking, you need not to do the blind docking. You can mark, you can prepare your grids at these positions. Okay, you can prepare your grids at these positions so that you can get the details information that it uh, is it interfering your active site or not, it is interfering your binding site or not. If it is so, it is interfering, then you know the function of this one, and accordingly you can interpret, right? So this will help you in this way. Okay, so we talked about various features of the protein. Now, these are the geo annotations. Okay, so the cellular components and its various terminology. So I am not going in details of this one, but if you have time, you can go through each one and you can explore a lot. So this is also very interesting area. Okay. Now, what are the enzymes and pathway databases that where your protein is interacting? See, you can get the details list of these proteins. So, a lots of secondary databases are linked. Okay, like Brenda, okay, Reactome. So these all are enzymes and pathway databases are linked here itself. That means if you uh, consider that your UniProt database, especially the UniProt KB, will be the your main gateway for your research. It will not be exaggerated. Okay, so you can consider this platform as your main gateway, because from here you will be diverted to other fields like the secondary databases, the structural databases, and all these databases, the ligand databases, and everywhere. The PubMed databases, all the databases are linked from here only. So this is the ideal path to me to start with. Okay, so you can think about that. All the research supervisors are also they can think about, they can guide their scholars that they can start with UniProt if they are working with protein information, whether it is sequence level or whether it is structure level. Now you will get some names and taxonomy. So you can see, so this is the your organisms. So what are the strains? Then what is the gene name? See why we got so many hits? Because we just typed the gene name only. Okay, so that's why we got so many hits. So by going through advanced searches, we can reduce the hit. Okay, so we can combine different queries and we can minimize the output. Okay, so these are the accession numbers for other databases. So these are the accession numbers for other databases. As I told you that several such databases are linked with this one. So now you see that I was talking about in the theory session that the subcellular location, right? So if you move your mouse, you see, you can connect this one. Okay. So if your protein is present in nucleus and what it is doing in nucleus, what is its function in nucleus, everything you will get it. And if you require the further mechanism, so you can go to the respective research paper. But you see that how they have means um, generated this information. So this is by similarity. Okay, this is by similarity. You can see this is by similarity. So it is manually curated. So your protein, this protein is present in all these cell organelles. So if you mark your mouse here also, you will get the location in the cell. Or if you mark the cell organelles, you will get the location. Okay, see, you will get the, see, this protein is not present in mitochondria. Can you see? So mitochondria, there is no protein in mitochondria. That means the presence of AGFI is not in the mitochondria. Okay, so otherwise they are present in these organelles. Okay, so now let's talk about some domain. You can see These are some topological domains. These are transmembrane domain. So many times we actually work on, say, transmembrane domain. So we need not to go for the structure of the whole protein. This is the point we make the mistake. Okay, what we do, we need the EGFR protein. We go to PDB and we get the full length protein structure. And that's the point we make the mistakes. So and reviewers are getting scope of asking lots of questions. So if I am interested in transmembrane domain, so you see that the membranes are listed here, so I can get the transmembrane domain. So I can get the sequence of transmembrane domain, and this is the sequence. So I can ensure the partial structure where this sequence will be included. Okay, and these are the sequences I should mark. These positions I should mark while docking. Okay, then effectively you can explain your data. Okay, so similarly you can go for other domains also. Okay, now you can see that the phenotypes and variants. Okay, so if the protein is uh, having some variants classes, so it will be listed here. Okay, now the post-translational modification. You see, there are lots of post-translational modification. Here you can see there is a huge number of, see, you can see the huge number of modifications. So if you want to retrieve this information from other primary resources, it's almost impossible. If you go for PIR, you can visit other protein sequence databases like RefSeq and of course in NCBI, including NCBI. So it's very difficult to point out information like this. So here you can see that disulfide bonds, glycosylation, okay, and all these modifications were done. So I can list if I am interested in signal. So this is the signal modification. So these are the sequences, okay, and the position is this. Okay, so these are the signal sequences. Okay. So if I am interested in chain, so these are the chains. So these are some chains of the protein, that is 25 to 12, 10. So these are the position of chain and this is the ID. Okay, if I am interested in disulfide bonds, see, these are the disulfide bonds that is present in my protein sequence. You can see so many disulfide bonds are there. And what is the positions? See, 31 amino acid at 30, position 31 has been formed disulfide bond at the next amino acid, that is the at position 58. Okay, now you can see because of three-dimensional folding, the formation of disulfide bond within the chain, within the polypeptide chain. Okay, so so many disulfide bonds are there. See, you will get the sequence accordingly. Okay, so we have two sulfur containing amino acids. So these will be used for the bonding of hydrogen disulfides. Okay, so these are the disulfide bonds you can test on. Okay, now you can get the details of this post-translational modification. Okay, you can go through this information and you will get the details of this one. Okay, so now you can see some proteomic databases. Okay, these are the proteomic databases, post-translational modification databases. That means, see, if you are going through means UniProt, it's your life is very simple. You have got the relevant information of modifications, and from here also you will be connected to the other secondary databases that deal with post-translational modifications. So you can go for further details. That means If you are interested to work in this field within few months, you can get all the known available information in this field, and you can prepare yourself and you can plan your research accordingly. Fine. So these are the means advantages of using this database. Now the interaction sections, I told you, this is the interaction sections. So these are the proteins that actually interact with several other proteins. And these interactions, you know that we can means predict the interaction by through string databases also, right? We can get that. So similarly, in this same window, you will be able to go through the all the interactions with other proteins, and that must be supported by your protein in your publication. So in some of these or any of these information or means interactions you are interested in, then the details mechanism of that interaction you can get from these manuscripts. Okay, so there will be some binary interactions. So these are some interactions, these are some protein-protein interaction databases. So for further details, you will be directed to with this string is there also. Okay, just I was talking about string. So they are there. Now the one of the most important one is the structure. Right? This is the structure of EGFR protein. Right? So this is the structure of protein. Okay. Now you see that now we will solve our that question that is my protein known or structure is known. Here you can see that your structure is not known. How do I know that? So if you go to databases, PDB databases, you will see lots of heat. But here you see that, okay, the model confidence and model color scale, it will give you the low regions. Okay, so these are the low confidence regions. These regions have been predicted very low with low confidence. You see that this is the alpha fold structure. That means here you can see it is a predicted structure. Okay, it is a predicted structure. So it is alpha fold only. If so, in the same EGFR protein in the demo session, in the theoretical session, we are discussing from human. And you know that we have got the structure from human, several structures are there in the PDB databases, right? If you remember that one, just a minute, I will show you the structure. See, this is the same protein, but from say humans, and you see, this is the structures. So there is a series of structures, not only these three. There is a series of structures. Then we get confused that which one to select, right? See, in these cases, you have to select the structure of your interest by going through the details that we discussed, because you might be covering some active sites, you might be covering some domains, so that must be included. Here you see that this is a PDB structure where you see the amino acid positions for these structures were solved by X-ray crystallography from the position 695 to 1022. Okay. Now, say I am interested in a particular binding domain, and that binding domain falls under this sequence region. Then I will select this one with this PDB ID. Then I will go to PDB database. I can put this PDB ID and I can retrieve the details information regarding this protein, or in an alternative way, the link is given here. I can get the sequence from or structure from here also, but I am not going to select this one. that is 1 NQL. You know that PDB ID are full alphanumeric numbers, right? So these are the PDB ID. So now here you see that there is a sequence where 25 to 642 six positions has been solved by X-ray crystallography. But my domain of interest is there within this one. So I will not go for the whole sequence. The sequence length is say 1200 plus. So I do not need the structure of 1200 plus amino acids. So this is you must be careful about this one. So people are interested in 1200 plus structures. If they are not getting that structures, what they do, they make the model or they predict the structure. So they use either predicted structures or they means go for homology modeling. And both the cases, they actually compromise with their quality of structures where the structure has already been solved in the databases by extracrystallography. So in those cases, definitely you will go for the structures that is this one in these cases. Now you think about another case, the case 2. What are the case 2? See, if my structure or my target domain falls under these two categories. Okay, the two structures is available. This is one MOX and one NQL. These two structures is available. Now, which one I should select? Okay, and both the structures means sequence. Actually, both the structures is encompassing my target area. My target area say 300 to 450 amino acids. That is my target area. For assume that one. Okay. So both the target area is my target is covered in both the sequences, both the structures. Now which one I should select? So I should select for higher resolution. High resolution structures I should go for. So which one is the high resolution structure? So you might say that 2.8 is the high resolution structure. This is wrong. This is wrong because high resolution structures means lower magnitudes. High resolution structure means lower magnitudes. That is 2.5 is comparatively lower magnitudes as compared to 2.8, and we will say that this one MOX is having higher resolution as compared to this one, that is 1 NQL. Okay, so in these cases, we will select this one for our further study. I hope you got this point. Okay, so here in this sequence, there is no structure. So now you can ask me that then what should I do? I should go for structure prediction. So definitely we have to go for structure prediction, but not for AlphaFold. So first try to build the structure by using homology modeling. Okay. If homologous sequences are available and the structure of homologous sequences are known, then people should go for homology modeling to build the structure. If not, then only they can go for AlphaFold because AlphaFold actually applies the fold recognition algorithm. Okay, fold recognition algorithm. So that's why the accuracy of AlphaFold comparatively lower as compared to homology modeling. So our option one is the X-ray crystallography or NMR structures from the PDB databases. Option two, if protein structures is not resolved by these experimental techniques, then we should go for homology modeling, right? If option two is not possible, that means homologous sequences are not available or the structure of homologous sequences are not known, then you cannot go for homology modeling, then you have to go for AlphaFold. Okay, so AlphaFold is the third option. Okay, and all these things you have to write in your manuscript. Okay, otherwise reviewer will ask you questions. So why you have chosen homology modeling, or why you have chosen AlphaFold? So everything has to be justified. Okay, so these are the domain. So this protein has these domains. You can see there is a domain that is protein kinase domain. So now you know what the kinase is, what the kinase does. So definitely the kinase will transfer the phosphates from one molecule to other. So there you go the interactions. That means to which protein it interacts. So now you can connect the interaction segments. Okay, so you will get this information and you can connect with this interaction section in which protein it interacts with, and now you can see that very important one, it has the disordered region. So if this structure is solved and this structure is excluded, the disordered regions must be excluded, because the disordered regions, if you your sequence is having the disordered regions, that might play with the accuracy of your interpretation. Okay, so that's why you must be careful about the disordered regions. So earlier I was talking about the disordered regions. So you must look for disordered regions and you must note the position of amino acids of the disordered regions, right? So these ordered regions should be excluded while you are solving the protein structures. Okay, so these are some important domains, and you can go through the functions of those important domains, and accordingly you can have your research plan. So what is your plan with this protein? Which function you want to target? Which domain is responsible for that function? And accordingly you select that one. Okay, so these are the phylogenomic databases you can go through. So these are the so many databases. You can see so many databases. Okay, so tomorrow and day two we will see few of them only. Okay, so when you get time, you can explore these resources. So these are very important resources, and definitely they are going to help you in your research. Okay, and finally we have come to the sequence segment. Okay, so these are the sequences. So many times people actually copy the sequences in this way. So this is not the right way. You will get lots of spaces, and if you use these sequences for further analysis, you will get lots of error. Okay, so you just go to these sections. This is the copy sequence, right? So you copy the sequence, you see the sequence has been copied. Now you can open the notepad terminus and you can copy the sequence. You see, this is the sequence. Now you know how to convert it into FASTA format. So you just add the greater than sign and you just mark as per your requirement. Mouse, okay, and you save this one. See, I am saving here as a dot pasta, and make sure that this one is in all file format, not in the text format. Okay, so now you have saved your sequence in FASTA format, and that means you can utilize this sequence for your further research. Okay, so that's the way how you get the sequence. Okay, so now if this is your sequence length. So from here you get the length of the sequence, and accordingly you can filter your protein structure. So which protein structures you should consider for your research. You can select from here. Okay. So these are isoforms I was talking about. So there are two isoforms, but remember they are computationally mapped potential isoforms. They are not verified one. So if you are working on this, you should be careful. Careful means definitely you can use this one. That doesn't mean that you cannot use this one. Definitely you can use this one, but you have to study in detail so that any questions from reviewer side can be addressed. That you should keep in mind. Okay, so these are the entry names. Okay. Now, these are the sequence conflict. So you can see that in various experimental methods, in this position, 539 positions, few techniques suggested that there should be C, and in some there should be W. So this is also an important information. So when you are actually analyzing something, then if you are facing this type of confusion, then you can means get the reference of this one and you can justify your output. You can say what I am saying, that if at 539 position your molecule is binding better with C as compared to W. That means this is a conflicting regions, na? So some people has reported that C must might be there also. So maybe the X-ray crystallography has made some mistakes and they have wrongly incorporated W. So accordingly, you can justify your data, right? So that's what I was talking about. See, these are the regions of conflicts. Okay. Now the sequence data for nucleotide sequences, you can get the nucleotide sequences, the gene sequences from GenBank, EMBL, or DDBJ. You see, you have the link for all the three major primary databases from here itself. And similarly, for protein sequence, you can get it directly from here, or you can get the sequences from here also, and you can compare the sequences also. If there is any positional biasness or not, that must be compared, right? So from any of the databases, you can get the sequences and it must be computed because you see they are mRNA, right? So mRNA might be changed, might be modified. So it should be compared. Now you see that the similar proteins, so these are the in mouse, they are 100% with 100% identity. You see, similar proteins with same protein, right? With 100% identity. So that means there are some data redundancy. Okay, so you see that this length is only 29 amino acids, and this is 24 and 18 amino acids. That means they had some different objectives. Okay, they had some different objectives of these proteins. So these are the, you see, the unverified one. So definitely we will go for this one. So currently we have selected this one for our research. Okay, so that's all about means UniProt. If you have any doubt, I can clarify. Okay, otherwise we'll move for the next one. That is the protein data bank. Okay, any queries? You can ask me. Okay, if not, then we can move for the next database, that is the structural database. So already we have discussed in details about the sequence databases. Now we will see that how we can efficiently manage the structured databases and what it means, right? So you see that uh, our plan was to go to structured database, but via UniProt. And I also discussed with you that why I prefer means protein databases via UniProt, structured databases via UniProt databases, because it's give me the accuracy of selection of the correct protein. So otherwise, many times we can work with the wrong protein. Okay. So, if we now discuss the protein data bank, the structural bioinformatics resource, you see that this protein data bank is a global repository of 3D structures of biological molecules. So it's having not only protein molecules, okay, but also the other molecules like structure of DNA, structures of RNA, and other complex structures, that is DNA RNA complex, protein ligand complexes, okay, ligand DNA complexes. So these all complex structures are also present. And it was established in 1971 and maintained by Worldwide, okay, PDB consortium. Okay, this is maintained by them. Now, why we use PDB? So it provides, this is the only one database actually that provides us the atomic level details for understanding biomolecular functions, right? So this is the only one database that provides us the atomic level details for understanding biomolecular functions. And that is essential for drug discovery, structural biology, and molecular modeling, right? So without structure, we cannot go for drug discovery or molecular modeling. So it's the structure you will use in computational docking, the homology modeling, and other bioinformatics analysis, right? So that's why it's the very, very important source of information. So now all of you know, all of you have used this terminus for retrieving sequences from PDB. So basically, today we will focus on how data is organized in PDB, how we can get the information from PDB, and how we can handle errors in PDB structures. So always there are errors. So please do not think in that way that whatever there is databases, those are 100% accurate. So some errors, but if it is reviewed one, the error will be minimized. If it is non-reviewed one, then there will be chances of error are more. Okay, so that we need to understand. So similarly, in structured database, there are also errors. How to handle that? Many times, whenever we do docking, simulations, okay, other analysis, we see our software is not working. It's giving error. We do not understand that how or why I am getting these errors. Okay, so today I will give you one example so that it will make you think whenever you get some error that where the errors are, so accordingly, you can make your back study and you can complete your job. Okay. So the structures that is present in PDB are from three sources. One is from the X-ray crystallography is one source. Sorry, yes, the X-ray crystallography. Then NMR. Okay, NMR is another source, and cryo-electron microscope. So these three techniques are used to solve protein structures and are deposited in PDB databases. So PDB files are very, very important to understand. Okay, it's a very complex files. It's very difficult to understand. Still, today, few of us we actually understand fully the PDB files. Okay, so you know that these 3D structures they contain several sections like headers, titles, queries, atom, each atom. So what are those actually, and what are the specific columns actually signifies? That is very very important. Okay. So please go through the PDB manual if you get time. I know it is difficult and it is boring, but once you can go through it, you will be expert in data and management in PDB. Okay, and so these PDB structures will be used by various tools like PyMols, Chimera, okay, for uh, structural analysis. So you see that header file that actually provides the metadata, okay, metadata of the file. The title will give you the full description of the structure. Sequence, one of the most important segment that will list your protein sequences in three-letter code. Okay, just previously we used most of the cases one-letter code. No? So this is the three-letter codes. The atom is a section, okay, that contains atomic coordinates, the x, y, and z coordinates of all the atoms. Okay, one amino acids will be composed of several atoms. So each of these atoms except hydrogen, each of these atoms except hydrogens, their positions in terms of x, y, and z coordinates will be deposited in the atom section, right? So this is actually deal with the 3D structures of protein. Now heat atom. What is the heat atom segment? So it describes the non-protein molecule. That means say your protein, this is your protein, it is actually bind with chlorine. Okay, that means this is the heat atom. So its coordinates, its position, where it is bound, which is the position, it will you will get in the heat atom section. Why it is important? So you see already chlorine is bound there. That means if you are designing a ligand, if it can bind in the same position, that means it will bind competitively for chlorine, and it can give you more insights of the molecular function of the protein. And the connect files will define the bonds between atoms and important for ligands. So this is the partial structures of PDB file. So you know that this is the header section. So all the PDB file, it starts with header, right? So it will give you the metadata of the file. So it is the date of the deposition and say this is the ID, PDB ID. Okay. Now you see that this is the title. So it can be a single line or multi-line here field. So here you can see the compound. So this is the complete description of the compound. So this is an enzyme with enzyme number. Okay, if it is the engineered protein, you see it's yes, it is an engineered protein. So in this way, you have to go through each and every detail. So if you open up a PDB file, I'm sure you have experienced this one. It's a huge file. It's a huge, right? So it's very difficult to go through each of the segments. But that's why the selection of protein is very important. So you cannot randomly pick up the proteins and you cannot go through repeatedly with this file. So if you take the correct one, you can save your time, and whenever you get the correct one, you can go through the PDB files in for details understanding. So there will be the remark section, which again is the important one. So in this way, there are several segments. I have not mentioned those one unnecessarily. The session will be means lengthy. So that is not our objective. Our objective is to tell you what are the important segments, what are you should focus on, and you can go through the PDB manual, and each of these sections you should understand column by column. So which column means gap, which column, which number indicates what. So everything you should know. Okay. So these are the softwares that allow us for visualization, and these are the applications of means PDB. You know that for structural bioinformatics, drug discovery, comparative modeling, and mutational analysis. So these are the only one source we have. So just I was talking about that is the PDB file I can means use uh, by closing our eyes? No, this is not possible. Just look at this one. Okay, so see, these are the atom number. You can see these are the atom number. So this is the atom section. Okay, so these are the atom number, this is name of the atom. Okay, this is name of the amino acids. So these atoms are from this proline amino acid, right? So this indicating the chain. So that's why actually I was asking that you should go through the manual and each of the column. So what would be the length of this column? So everything. So this is also informative. So again, you have to understand from the file. Then only you will be able to write your manuscript in a efficient way. Okay, so this is x coordinate. Okay, this is y coordinate and this is z coordinate. Okay, this is the connectivity and other important data. So what is the atomic type? Nitrogen. See, this is carbon. So this is also carbon, but this is 3 alpha carbon, right? So in this way, each of the column will give you different information and you should know the length of the column. Okay. Now what I was talking about, see the error. See, this is the error. Where is the error? Now you see at atomic position, this one, this position, sorry, this one for the amino acid. See, you see AGLN, right? That means this is glutamine amino acids. Rest are fine, na? Rest are fine. But what is here? You can see this is along with the name of the amino acid. Some extra letter is there. A. This is B. This is again A, B. That means there are some errors. So when your software will read this file, so it can read this one. This is proline, right? But there is no such amino acids which has four letter code and that is AGLN. So it fails to read this one. It fails to read this one and it will show you error and you will be in a difficult position to find this one because it's very difficult to know that this sort of error because it's a huge file. So where this sort of error is there, it's very difficult to locate, right? So that's why whenever you find some error, you must check your file. Okay, that's why we should check every file before talking. It's not like that we will get the code, we will download the file, and without going through the file, without pre-processing the file, we will use for any further analysis. That is not right way. Okay, so that is the error from this one. So what are the key takeaways from day one? So we have gone through the introduction of bioinformatics databases, understanding their biological role in research. Then we also discussed about exploring UniProt databases, and we also discussed about PDB databases. Okay, and we have seen that how we can search, filter, and extract relevant biological information. So that's all for today. And thanks for listening with great patience because it's a lengthy discussion. So I will appreciate if you ask me questions. I'm open for questions. Any questions are there? Okay. Thank you. Thank you for participating.